It's a better cleaner, is it? Okay, that's a useful tip. Thank you. Thank you. Thank you.
Welcome to today's session. We're going to have a pair of lectures by John King, University of Nottingham. First lecture will be simple three boundary models for tissue growth.
Thank you. Thank you. So I think these will be rather different, both in style and content, from most of the talks here. So I hope the variety is not unhelpful. Shout out, please feel free to shout out and interrupt. I'll be facing the board some of the time, so do shout. And I am still a bit jet lagged, so that would help keep me alert. Thank you. So I'm talking about tissue growth. So today I'm going to set out what I regard as some of the basic free boundary models for it. In particular, how you incorporate some of the simplest biophysical processes into them. The second talk today will be multi-phase modeling, so a different modeling approach, and that'll be slightly more abstract. The final talk tomorrow will be, in effect, a PDE talk. So I'll extract in the previous two talks a fairly generic framework which has a lot of the relevant structure in it and do some formal analysis of it tomorrow. So why did I get into tissue growth? So the earliest work I did was on tumours, as is traditional in this area. Actually, a lot of the early work was. Subsequently to that, we had a strong group of tissue engineers at the university. So I looked at growth of tissue in the tissue engineering context, which is different because the stuff's growing out of the body. Around similar time, I was talking to our bug people. So biofilms, although they're made up of bacteria rather than mammalian cells, they have a number of properties in common. So some of the generic modeling statements go over. And then I guess the big endpoint, developmental biology, one might come to is developmental biology. So how would you grow an entire human out of a single cell? How would you do that mathematically? That's way beyond anything we can do yet. But it does motivate, in particular, the multi-phase thing, because we're all made up of lots of different cell types. So if you're going to do that with continuum modeling, you need multi-phase setups. Okay, so that's the big picture. I'm going to start with the simplest models of all, which are ODEs, because they give you some key pointers towards how to deal with what you need to include in the PDEs. So let me start with Gompertz, which has nothing to do with anything else I'm going to talk about, but is a phenomenological setup people often use to assess growth rates of tumours against. So the Gompertz ODE would be something like the following. It's a slightly odd choice of nonlinearity. So S infinity is the final size the thing gets to. S is the radius of the tissue. So a lot of the talk will be about the spherical case. Okay, we've heard the standard joke about spherical cows already at this meeting. Remarkably, in vitro, often these things do grow as spheres, but stability is important as well. I'll say a few things about that. So tumor spheroids, but also more recently, organoid bodies and things are in vitro cases, stuff growing outside the body. Depending on the cell type, they may be spherical, they may not. You may have seen stuff about brain organoids where you start to see the very complicated structure you'll see in brains, which is very far away from that. So why am I telling you this? Well, the growth curves, if you solve that ODE, you write down the Gompertz function. Initially, if you start off much smaller than the saturation size, it will grow exponentially. And that can be understood asymptotically as a limit. There's an intermediate phase, which is near linear growth, which isn't an asymptotic limit of Gompertz. So I'll come back to how you get it as a limit. And you might want to think about why it looks so straight if you plot Gompertz. But then it does turn over and saturate. So if the thing grows to a finite size, you end up leveling off. So there are three phases. And the things I want to build into are both the ODE versions of that thing, what the mechanisms underpinning it are, and then how you reformulate that as PDEs, as free boundary problems. Okay. So what am I going to have? I'm going to have a number of different variables. So N will be the number of cells in the growing tissue. Correspondingly, V will be the volume of the tissue. A will be the surface area. And the thing I've already introduced is S as the radius. And I'm going to straight away split it into 3 phases because I think it is instructive in terms of how you then set up the PDEs. So the first phase is nutrient rich. So the cells all have an ample supply of nutrient. So they're all dividing and dividing at pretty much the same rate. So the model you write down is Malthusian growth. So the production rate of new cells is simply a constant times the number of cells because they're all dividing. If you assume cells all have the same volume, that's equivalent to saying dV by dt is K times V. And I'll put a box around that because I'm going to compare it with something else in a minute. And then if you write that in terms of the radius, the volume is proportional to radius cubed. So you get an extra factor 3 that I'll take over to the right. So the growth law for the radius looks like that. Okay. And that's pretty much all you have to say about that first case. It is exponential growth. So S is the initial size times e to the kt over 3. And that corresponds to the first phase I've hinted at the picture above. So the second phase, you start to worry about nutrients. So how do you get to linear growth? Well, the most established viewpoint on that is it's nutrient limited. So if I do my spherical cartoon, there's the tissue. Nutrient is diffusing in from the outside. It's being consumed by the cells around the edge, so it doesn't get to the middle. So the PDE version of this is more complicated. But if you're to write it down as ODEs, then you're going to have the cells dividing at the same constant rate out there, but not dividing in the middle, because then either nutrient poor or simply dead. And the simplest assumption is to take the thickness of that dividing rim to be some constant L. Okay, so sticking with S for a minute, that means ds by dt. The rate of change of volume gives you an S squared, and then I can cancel the 4 pi from both sides. So ds by dt is essentially the volume dividing ones, which is the total volume minus the ones in the middle. So that's going to be a third K times the total volume, which is S cubed, minus the ones that aren't dividing, which is S minus L cubed. Okay. So initially, that requires S to be bigger than L. So initially, you're in the picture above it, but then you start to experience these limitations to do with lack of food supply. But the most important thing to understand about this is when S is much bigger than L, you just expand the right-hand side, and that becomes as a 3 from the cube. So it becomes KL times S squared. So the S squareds from the two sides cancel, and you get a linear growth law rather than an exponential one. The more revealing way to then rewrite that is the growth law is now no longer a volumetric one, but an area-based one. So it's KL times the surface area A. So one aside on that final, well, I should have put a box around it. One aside on that final formula is that sometimes people have, right, it does hint at what an instability mechanism might do for you. Okay, so a spherical lump of tissue has the smallest area for given volume. So you can make the stuff grow faster by not being a sphere. And there have been some proposals to do diagnostics based on fractal dimensions of melanomas and things. Okay, so the edges are very complex. So if it really were fractal, the area doesn't scale like S squared anymore. It's some power larger than 2, which means you'll get a superlinear growth rate. But the physical statement embodied in that area relation is the main thing I want to highlight. So that's second phase, that's linear growth. And then the third phase is growth saturation. And possibly surprisingly, it's not entirely clear how that works because you're making new cells all the time around the edge and you've got to get rid of them. But it's only the simplest setup that'll do it. So I draw a similar picture to before, but now I'm remembering that the rim is much smaller than the rest of the tissue. The situation is something like that, and I'm now going to have the stuff in the middle dying. So that's minus KD, the death rate, and K for the growth rate still for the rim. So saturation ends up being a balance between those two effects. Okay. So that time it's going to be S squared ds by dt. If I keep the second version over there, there's a KLS squared. And then these ones were killing off the stuff in the middle and almost everything. Almost the entire tissue is the dead stuff. So that's S cubed again, the thing scaling with volume. So that ends up giving you your growth law in the final case, dv by dt. I may have dropped a factor 3 somewhere, but I'm not going to worry about that. There's the same growth law as before from the cells dividing around the rim, but now you're killing them off at a rate that goes with the volume. Okay, so that's the final geometric version of the growth law. So the statements about S are all very specific to the radial case. The other statements have slightly more general truth in them. But in particular, from that S equation, you'll see the thing goes to a finite saturating size. Okay, so that's ODEs, and I'm now going to build each of those into a PDE, free boundary setup. But any questions before I move on to that? Okay. So PDEs. So now things need to depend on X, as, the spatial variables as well as time. So the thing I'm going to replace capital N with is N of X and T, which is the volume fraction of cells. And we're going to need other extra variables shortly. So in particular, and importantly, N's got to lie between 0 and 1. Which rules out the very first thing you might want to do. If you look at the first equation for capital N and just do a direct analogy, I mean, it's fairly obvious it's not going to work, but the thing you would write down is simply that. The cells are all dividing, so the overall growth rate is going to be given by K times N. In overall terms, that's still right in the first nutrient-rich case where all the cells are happy. The growth rate has to be exponential because they're all dividing. But this thing clearly won't work because of this constraint. So the next thing you might attempt to do is have the cells move around to stop, to spread themselves out a bit, but that clearly won't work either. So if you write down the linear reaction diffusion equation and solve it, you've still got N growing exponentially. So that fails for the same reason. So possibly, unsurprisingly, one of the things that gets done is to go to Fisher instead to saturate the growth rate. Okay, so it tends not to appear in a model that's simple. It tends to be embedded in much more complicated models. I don't like it because the saturation term, you've got to ask what it's for. The cells are all happy, so they should all be dividing. It's not like the ecological context where you're competing for food. So it's very far from clear what that term is supposed to mean. And I'm going to rule that out for that slightly subtler reason. Okay, so you'll find what I do is I advocate against using reaction diffusion equations and then come full circle and start analyzing them because I'm going to put different content into them. So that's where we're headed. And the final talk will be a reaction diffusion equation. So what do I do instead? Well, the thought experiment is if a cell is dividing in the middle of tissue, it can only do it by pushing its surrounding cells apart. So the only sensible way to proceed is to include a velocity field and write down the conventional continuity equation in terms of that velocity field. So you think of the thing I've just written down as a time derivative, but it's a time derivative in a deforming medium. So if you use the product rule on this combination, there's two terms. There's the v dot grad n, which says the stuff gets convected with velocity v. But there's the end V thing, which says if the flow is in, the volume fraction is going to get diluted. So again, you can think of that as the right time derivative, but it's got effects in that certainly aren't there. And that's going to be K times N. Okay, so that's all well and good. But of course, I've introduced a whole set of extra variables and I've still only got one equation. Right, so in D dimensions, I've got D plus 1 variables, the components of V and N. So I'm certainly going to need extra equations. And I'm going to build them in 2 steps. But before I do that, let me be explicit. We are now in free moving boundary territory. So that equation is going to be satisfied on the tissue domain. I'm going to have, in most of the things I say, there's going to be no cells outside. So n is going to be greater than 0 in there, and n will be 0 there. And the other notation I'll need is the normal velocity of the moving boundary is q nu, and the normal direction is nu. So I'm going to write the unit normal vector as nu hat. Okay, so this is going to be essentially appear in the rest of this first talk. So will the second condition, with one exception, which is the kinematic condition that says that the edge of the tissue grows with the tissue velocity. So the normal component of tissue velocity is v dot nu hat. So that's one of the free boundary conditions. Okay. So the things we need to solve for are n, v, and the free boundary. Okay, so we're lots of equations short, and I'm going to build them back in by going through the same 3 steps I had in the ODE case. Okay, so the first of them is exponential growth. And I'm going to break it into 2. Okay, I need to keep track of time. Yeah, I think I'm going to break it into 2. The first case is to add 1 extra equation by making the cells close packed wherever they exist. In other words, the volume fraction is going to be n is 1 inside the tissue in gamma of t, omega of t. Okay, so that's 1 extra equation. So that's going to be enough in 1 dimension. It's not enough when you go to the fully higher dimensional case, but I will pursue it with radial symmetry. So then the continuity equation just collapses to div of v is equal to k. And in the radial case, then I can solve it. So radial in 3D, the only non-zero velocity component is in the radial direction. So if you remember how divergence works in radial coordinates, you'll end up with v as a third kr. And then this equation tells you how fast the thing is growing. So it's spherical. So q nu is just going to be s dot. And then the velocity field on the boundary is a third k r, and r is just s. So we get back to exactly what we had from the ODE. So I'll give myself a tick there. So the radial case is done. We're back to having the right number of equations as unknown, but the thing might not start off radial. We're going to need to include extra biophysics, if you like, in the non-radial case. So tell me if you can't see what I write over here because of the bias. So non-radial, it immediately tells you we need a constitutive law. We need to say something about the mechanics. So far, we haven't needed to. I'm going to do 2 cases. Okay, the simplest. So traditionally used in tumor modeling, I don't entirely approve of it in tumor modeling because it's not growing into a porous medium, but in tissue engineering context, you do grow tissue in vitro in a porous scaffold is a standard technology. So that says it's Darcy's law. So the velocity field is flow down a pressure gradient. Kappa is to do with the permeability. Okay. So that's Darcy's law. So if I rewrite, if I stick to this close pack case where div v is equal to kappa, we've got a Poisson equation for the pressure field. I'm taking all these material quantities to be constant so far. So that's the equation for the pressure field. That's holding inside. And then on the free boundary, we're going to have the normal velocity is again given by the velocity field, so kappa times the normal derivative. And then the simplest thing to say is if you're growing the thing in vitro, you just got some growth medium outside with no mechanical stiffness or viscosity. So take constant pressure field. So that's the simplest bona fide multi-dimensional free boundary problem I'm going to write down. So it's a famous one in other contexts. So there's a bit of biology in there that the stuff's growing. You don't have in most physical applications. But there's a thing called the Healy-Shaw squeeze film, which is a two-dimensional model. So you stick a fluid between two parallel plates and then squeeze the plates together. And the fact that you're squeezing the plates together pushes the flow out. And that mathematically ends up being equivalent to this biological growth term. Moreover, beyond the fact that's known in physics, it has a huge number of special properties. Because of the Laplace operator in 2D, for example, you can write down lots of explicit solutions, non-radial ones using complex variable methods. So this is actually quite a famous setup. If I've got time at the end, I will talk about the stability of that. I will do the stability analysis. It's the only one I'll do. I'm going to make passing comments, though, on why stability analysis matters. And I've already alluded to one of those. So that's the close pack case. Going to higher dimensions, you get this free boundary setup. The other thing that might not happen is you might not start with the cells being close packed. So then you need to say something different. So non-close packed. So we're still going to have n being 0 outside, but inside n will at least start less than 1. So the simplest thing to do, it's formal, the simplest thing here is the following. Introduce. Right, so P is the pressure between cells, but it's kind of the incompressible scenario where it comes out as part of your solution. If the thing's not close packed, the cells essentially feel around to know how many others are nearby. So I'm going to specify cellular pressure as a function of n, and then use Darcy again. But this is then capital P of n because it's specified as a function of n rather than being an extra unknown. And then I'm going to write V by analogy is some permeability which will now depend on n times grad of P. So that'll give me a different moving boundary problem. So it'll give me a complementarity problem, which mathematically is the reason I wanted to mention it. So now here's our tissue again. I'm going to start with the stuff not being close packed, but because of cell division, plausibly it will become close packed in the middle. N is going to be between 0 and 1 in the surrounding region, and n is still 0 outside. So that's representative of the setup I'm now talking about. Okay, so that's going to be minus the inside bit, that'll be plus. So minus will be as above, so Laplacian of P is minus k over kappa, and then the plus part will be dm by dt equals div of kappa n grad capital P of n plus k of n. Okay. So that's the first time I've explicitly written down a reaction diffusion equation, and they will rear their heads periodically throughout. Okay, so different governing equations in the two regions, so I'm going to need the appropriate continuity conditions between them. So this thing is going to require P to be bigger than some critical pressure, which is the one at which, so P of P of close packed, capital P of 1 is going to be P star. Okay, so in the central region, P is bigger than P star and N is equal to 1. In the rest of it, P is less than P star and N is less than 1. So there's complementarity between those two. And then on the interface, well, on the outer side of the more dilute region, N will be 1. On the inside of the close pack region, sorry, on the edge of the close pack region, P will be P star. So they're fairly familiar setups. And then because that boundary is not known as well as that one not being known, it's a double free boundary problem. We need a third condition, and that is that the velocities be equal. So you have to be slightly careful deriving it. So kappa dp by d nu minus will be kappa n dp d capital to p by dn plus. Okay, so they're the kind of free boundary problems you get in the growth. Yeah.
The n, is it allowed to be both 0 and non-zero at the other boundary? Does it come from the equation?
So in this setup, because the boundary moves with the local velocity, the second derivative term is irrelevant on the boundary. So essentially you just get the ordinary differential equation. So n on the boundary will be n, but no, I've got to be careful, that can't be right. Right. I have thought about that question before, but the answer isn't in my head. Let me come back to you.
Okay.
Yeah. Yep.
What about K of n diffusion coefficient?
Mm-hmm.
What do you know about K of n?
So kappa would, right, but I haven't taken this kind of analysis beyond just getting to the setups, but kappa will be an increasing function of n, decreasing. Right, the larger n is, the larger the pressure gradient you need to push the stuff. So kappa will be a decreasing function of n.
What kind of nonlinearity? You don't know what.
No, I mean, you put in a pal or an exponential if you wanted to push it. Yeah. Yeah. Okay, so, sorry, was there someone else? Oh, okay, so the other constitutive law I want to talk about is Stokes, which I'm happier with in this situation of unconstrained growth where you're not growing the stuff in a porous material. I should add that the purely biological case, there is extracellular matrix which mimics a porous surround. And when I get to the multi-scale case, implicit in that will be stuff to do with Darcy, but for a slightly different reason. Okay, so the second simplest is Stokes. So the second simplest is to treat the growing tissue as a standard viscous material, so Stokes flow. Okay, so the inertia is certainly negligible. So it's there's no inertia terms. So there is now a lot of work on elastic and viscoelastic and stuff, modeling of tissue. It is much more complicated and there are additional issues. So it's not like standard elasticity. Because of growth, you don't have a reference configuration in the same way you do in standard elasticity. So going to elasticity, even the initial setup raises extra issues. And I haven't worked on that, and I certainly don't want to go there. So we need constitutive law, as I've alluded to get the right number of equations as unknowns. So in Stokes flow, the stress tensor is minus the pressure. So that's isotropic, so it multiplies the Dirac delta. And then at various points, I'm going to switch to 1D, because otherwise I end up writing down huge numbers of suffixes. But to keep everything explicit, mu is the shear viscosity. I'm not calling it mu because mu is going to appear with another reason in the next talk. So that's the shear viscosity term. And then because, although the thing's incompressible, the divergence isn't zero because of growth. So you do, in general, have to have a bulk viscosity term as well. Okay, so that's the standard constitutive modeling for a Newtonian fluid. So the momentum equations, negligible inertia, d sigma ij by dxj is equal to 0. And then the rest is the same as before, I guess. But I've got to tell you what stress conditions I'm going to worry about. So if I combine those two in the usual way, there's minus grad P plus nu Laplacian of V. And then you get two divergence bits because you get something from there. So nu plus lambda grad div V. Okay. So those are the PDEs which have to be solved within the growing domain, but with the incompressibility condition, which, as I've said, now is no longer the standard one. So there's a bit of biology on that right-hand side there. So that's, again, in the growing domain in omega. And then on delta omega, the free boundary, as always, the kinematic condition holds, the boundary moves with the velocity of the tissue. And then I'm going to do the simplest other thing, which again assumes the surroundings have no material properties. It's just a viscid fluid or whatever. So both the normal and the shear stress components are 0. Okay, so that actually looks a lot harder than Darcy because it's higher order and stuff. Fortunately, you can write down the general solution to this, which is slightly surprising, which means I don't need to do the stability of it because I can just write down the answer. And the answer is the thing just grows without changing shape. So the velocity field is radial, even if the domain isn't. So I can't remember how we noticed that, but certainly once you've noticed it, it's sort of obvious and you check it by sticking it back in. Okay, so V, the velocity field, is going to be, it's just the third K times the vector. Okay, so it is entirely radial, even if the domain isn't radial. There's no stress being induced on anything, so all the stress tensor components are 0. Because it's unconfined growth, it's unconstrained growth. And P is, my notes in a minute, is a constant. Is it 3 lambda plus 2 nu or vice versa? So there's a non-zero pressure inside when the pressure outside is 0. And you think of that as being this proliferation-driven pressure. The cells are having to push each other apart to keep on growing. And that's reflected in that expression for the pressure. Okay, so finally, I need to get how the boundary moves. So Q nu is minus, if I do it as level sets, and the boundary is going to be the 0 level set of capital F, then Q nu is that. And with this, the velocity field is given, well, let me write down what you get. So the level sets of F go like a third K x dot grad F is 0. Okay, so the first term is to do with this. The second term is the normal component of the velocity field written in terms of F. Okay, so you get a combination that we've had repeatedly before. The thing grows without changing shape, but grows with this rate, e to the third kt. So as soon as you add surface tension and things, you have to solve the entire Stokes flow-free boundary problem, which is much more complicated, and it will end up going spherical. I have got some pictures about this kind of stuff from Shekhar Venkataraman, but I'll maybe show them tomorrow if there's time. I don't want to keep flipping between the board and the computer. Okay, so that's the simplest Stokes flow case, and deceptively simple because you can just write down the answer. And that is the first of the three phases. So that's the nutrient-rich growth where everything is exponential. And any questions on that one? Okay, so there's two more to go. Right, so I should have time to do the stability calculation. So the next is nutrient limited. So as you might expect, I'm going to have to add an extra equation for the nutrient, but it does build directly on the things I've already been saying. Those ingredients all stay in place, and there'll be an extra equation. So nutrient limited. So C of X and T is going to be the nutrient concentration. So we want a reaction diffusion equation for it. It is diffusing, but it's being taken up by the growing tissue. Unfortunately, diffusion is much, much faster than growth. So I can write it in quasi-steady form, and I can ignore what would be the traditional left-hand side of the reaction diffusion equation. There's going to be some consumption rate beta, and I'm going to assume the rate of consumption is proportional to the rate of cell division, which is not a stupid thing to do and ends up giving you a nice simplification shortly. So that's going to be my nutrient consumption equation. The other equations are all the same as before, except C is appearing. So the growth rate is now going to depend on the concentration of nutrient. So Vn is K of C times N, and K is going to be an increasing function of C. The more food there is there, the faster the things are going to divide. So why have I taken the consumption rate to be proportional to the division rate? Because something fortunate happens, but I think I want to close packet first. Yes, I want to close packet first. So Darcy, again, the simplest case and close packed. Right, so the mechanical parts of it, Darcy or Stokes, are the same as before. So I'm only going to do Darcy because there's a big simplification which is about to occur. So N is going to be 1 inside the tissue. So the equation for C reduces to that one. And then the equation for P, as before, is the Laplacian of P is minus K over C of over kappa. So that is a closed free boundary problem as long as I specify the boundary condition on C. Okay, so C is going to be C star on the boundary. P is still going to be 0, and the velocity q nu is minus kappa dp by d mu. Okay, so that's the full free boundary setup. There are 2 governing PDEs now. So it's not as simple as cases we've seen before, and it's not as simple as the classical ones like Edie Shaw. But the reason I'm doing this, and in part the reason to take Dirichlet conditions on both variables, is C and P has a simple linear relationship. So I can drop it down to a scalar PDE setup. So I'm going to do it with K of C still general, and then I'll do the simplest case where K is a linear function of C, which is not a silly thing to do. So that's probably, I'll get my arithmetic wrong in a minute, it's probably kappa beta times P plus C is equal to the value on the boundary. I put the beta in the right place. Standing too close to the board, I can never quite see what I'm doing. Oh, sorry, I've forgotten the diffusivity of the, there's a diffusivity of the nutrient as well, which I'd inadvertently scaled to 1, which I don't really want to do because I want to keep track of it. So there's a diffusivity there and there. Okay, so linear relationship between P and C, and then we get down to a scale of PDE free boundary problem. So DC, Laplacian C, this doesn't change. That's exactly as it is above, but the whole thing is nutrient driven. And that's reflected in that being the governing PDE. On the boundary, C is C star. And now the normal velocity is to be rewritten in terms of C. So it better be dc over beta. No, sign change, dc by d nu. Okay. So that's the free boundary problem in the nutrient limited case in its simplest possible form. Okay, that if you run that out to large time, it does grow linearly. I'm not going to show that in part because I'll make comments about where you add additional effects, and it's in some sense clearer in the more complicated context. Okay, but I present that as a meaningful free boundary problem. And I'm not actually convinced very much has been done on it. If the right-hand side's not there, it's effectively Healy-Shaw, but it is a non-linear version of it. I will mention the linear case because that has an infinite number of conservation laws, like the standard Healy-Shaw problem does, like the Healy-Shaw squeeze film problem does, which underpins the comment I made earlier about the squeeze film case. So that thing, they are moments in the Healy-Shaw case. They're not classical moments in this one. And I'm going to copy them off because I'll get it wrong if I try and do it in my head. So the linear case, that is an increasing function of C. Division rate increases with nutrient level. And the things that are conserved are the following: the integral over the growing domain of phi, which I'll explain in a minute, e to the minus something to do with the growth rate, which is k naught c star t, that integrated over the domain is equal to 0. Any phi satisfying the modified Helmholtz equation. Okay, so for all dc, same equation as for c. dc, the Laplacian of phi, is beta k naught phi. Okay, so whether this has any connection with integrable systems and things, I don't actually know, but there are an infinite number of conservation laws. Okay. What was the next thing I wanted to say? Yeah, so that thing does grow linearly. The stuff in the middle, because it keeps on growing, the stuff in the middle is getting further and further away from the nutrient supply. So the stuff in the middle is going to die. And that forces you to think about having now two different phases. Even in the close pack case, I need a cell phase and a dead phase. So dead cells. So this is still the linear growth case. So dead cell volume fraction is going to be m. And my conservation of mass equations are dm by dt plus div of v times n is equal to, they're still dividing, k of cn, but now they're dying as well. So do that as capital K of c times n. So I've already said that k increases as c increases. Capital K is the other way around because the less food there is, the faster they're going to die in the simplest continuum assumption. So that's the first of my two continuity equations. I need a second one for the dead cell volume fraction. I'm going to keep it close packed because I'm going to add the two together to be one. Okay, so dm by dt plus div of v, I'll comment on that in a second, v times m. We're making new dead stuff by killing off the living stuff. So that's going to be k of c times n. And I'm going to maintain the no voids thing, the everything is one or the other inside the domain in gamma of t. Okay, so I'm doing multi-phase in the second talk. In some sense, this is multi-phase. I tend to think of it as 1.5 phase because I'm taking the same velocity field for both variables. Okay, so it is a continuum. They're both physically meaningful quantities, so they can move together unless there's something extra going on pushing them apart. So in this situation, but not after the break, we'll have the same velocity field in both phases. So what to say about it? Right, so the main comment I want to make at this level is that you get the loss constitutive laws, which I'm not going to do. Constitutive, I can't spell anymore. Constitutive, that's probably nonsense. That's constitutive law. I criticised putting the saturation term into Fisher earlier on as not meaning anything. I'm going to derive the same term here for a completely different reason. Right, so if you add these two equations together and use this no voids condition, we've got div of v is kappa c times n. And if then I stick that to, if I replace div of v in the first equation by this here, I'm not going to do anything with it, but I think it's a worthwhile observation. Multiplying out by the product rule, the divergent part, the bit I retain is the v dot grad n. So that combination is the fact that these cells are moving with velocity v. So their rate of change convex with that rate. And then the right-hand side, there's a k of c times n, which was already there. But then the n div v from the product rule there will give a factor involving n squared. So there's a 1 minus n there. And then there's a k of c times n. So you do get the saturation term for a completely different reason to why it's normally written down in Fisher's equation. And that, unless something odd goes on, is going to keep n below 1. So you're not going to get in the difficulties of n hitting 1, which I've alluded to, and will probably reappear after the break. Okay, so that thing leads to linear growth. But again, I'm going to jump straight to growth saturation because I'm not going to have time to do the stability. I'm going to jump straight to growth saturation because that captures this case as a special case, as a limit case. So I can do both things at once. So growth saturation. And one of the advantages in this kind of incremental approach to modeling is you don't have to do much extra work to add extra things. So there's going to be one extra term only, which is the degradation of the dead cells. So the M equation is going to change to the m by dt plus div of v dot m is the fact that you're making stuff by cells dying. And then I need to check what notation I've used. Yeah, it was KD again. And then the dead cells, so the subscript D, are going to be got rid of at a rate proportional to the volume fraction of them. So that final term I just written down is the only extra term. Okay, so what happens now? So N, I am going to jump in part because of the time, but I probably would have done it anyway. So N drops exponentially in the core. The things I'm now saying go back to this thin rim argument I used in the ODE case, that the rim of dividing cells is only a very small proportion of the tissue. So in the core, and the cells don't have enough nutrient supply, so they're dying off at a rate essentially capital K evaluated at 0, because C is exponentially small in the middle. So that means you can replace M by 1. So now we're in the opposite way round, that everything is dead and the dead stuff is close packed. If you track back through the various statements before, we just end up with that as our governing continuity equation. So exactly as before, a constant right-hand side, but with the opposite sign. So we're in slightly familiar territory, but that change of sign changes everything, in particular in terms of stability, and that's where I'm going to finish this talk. So exactly as before, set sign change. So in particular, if you do the Healy-Shaw reverse squeeze film, now reverse because the sign change, it's ill-posed. High frequency modes grow arbitrarily fast. So you certainly need a cutoff mathematically, but fortunately you get a regularization from doing the thin rim limit as well. Okay, but the presence of the rim does change the boundary conditions. So one does a formal asymptotic analysis based on the thickness of the rim going to zero, but it nevertheless having a non-trivial effect, changes the boundary conditions. So what happens for Darcy? We don't have kinematic anymore because Q nu is now the velocity based on the cell death, but there's an extra contribution to the velocity based on the division of the rim. So there's a Q plus V dot mu. Okay, so this is cell division in rim. So you can calculate the value of that quantity Q by doing essentially a traveling wave analysis. So the inside of the rim doesn't see the usual kinematic condition, it sees that one there. So we have the Laplacian of P is KD, subject to this condition. That helps a bit to stabilize because it's adding some growth and it's the shrinkage which causes the instability but doesn't do the whole story. Fortunately, that thin rim limit gives something extra as well. So a regularizing quantity epsilon times Q nu. Okay, so again, that's not imposed phenomenologically to make the mathematics work. It comes out of doing this asymptotic analysis. So in the Stefan problem literature, that's called kinetic undercooling. It's something completely different here. So it doesn't kill the instability. You do get fingering. And again, I've got pictures which I may or may not, from Vanessa Stiles, which I may or may not put up tomorrow. But it does make it well posed, but very strongly fingering setup. So Stokes, what's the corresponding statements? Q nu, again, you've got the same, right, that I should have written that in terms of the pressure, which I haven't. So I'm now just repeating myself. So kinematic condition as before. And then the thing that happens to the boundary conditions is slightly surprising. You get a negative surface tension. So the pressure, instead of being zero, is proportional to the normal velocity of the free boundary. So that, of course, the most common regularization is surface tension, but this appears reasonably often as well. Yeah. Okay, so I can't remember my sign convention. Let me write it as gamma, then the mean curvature of the interface times nu I. So the stress condition changes from being zero stress because you don't care about the outside to this thin rim having a non-trivial effect. And what's going on in the rim is the cells are dividing, so they're pushing against each other. So the rim is under compression rather than under tension. So you get negative surface tension. Okay, so Stokes with a volume loss rather than a volume gain is not ill-posed. Once you do the asymptotic analysis, you get the opposite of what's happening with Healy-Shaw, where the rim helps regularize it. That makes it worse and probably actually makes it ill-posed. But the numerics run out to a certain time at least. So that you get this viscous buckling in the rim. The compression of the rim causes a buckle, and that causes an entirely different type of fingering instability. So that's the most interesting instability in the Stokes-Slope case. Okay, I will finish there. Thank you.